Sitting at the center of the brain, the midbrain, also known as the mesencephalon, is located above the hindbrain, below the forebrain. This region differentiates from the forebrain and hindbrain quite early in development. 3 to 4 weeks after conception, when a fetus's central nervous system is just a closed tube of cells, these three structures are visible as three clear bulges, the first segmentation of the nervous system into functionally distinct regions. The midbrain is the middlemost bulge, or vesicle. This soon changes as the midbrain folds in on itself and the forebrain and hindbrain expand around it, completely encasing the midbrain. The midbrain of humans is remarkably similar in its structure to the midbrain of most vertebrates. The midbrain is associated with vision, hearing, and motor control. It's also associated with the regulation of sleep and wake cycle, arousal and alertness, and temperature regulation. Like virtually all brain regions we'll study in this unit, the midbrain's structural divisions indicate functional differences. The tectum, Latin for roof, makes up the top part of the midbrain, and it controls the auditory and visual reflexes. In humans, the tectum consists entirely of 4 small bumps, the colliculi, or in Latin, little hills. The colliculi come in 2 stacked pairs, the inferior and the superior colliculi. The superior colliculi, the uppermost part, are devoted to vision. They receive inputs both directly from the retina and from V1, and control an array of visual reflexes, especially those involved in tracking objects moving through the visual field. They exert this control by stimulating ocular motor neurons. The motor movements of the eyes can be initiated voluntarily, but they also occur reflexively. This adaptive, automatic orienting allows you to continuously adjust to changes in your visual field. Interestingly, you also show this sort of midbrain-generated eye movement during the phase of sleep called REM. The other pair of colliculi, the inferior colliculi, are involved in auditory processing. Like the superior colliculi, the inferior colliculi modulate sensory orientation in a basic reflexive manner. They control, for example, the startle response. The reflexive responses that the midbrain generates allow for adaptive, evolutionarily crucial reactions that the cortex is far too slow to carry out. The midbrain is also directly involved in a variety of motor processes. This role involves the region of the midbrain known as the cerebral peduncles. These stem-like bundles of nerve fibers help transmit motor impulses between the neocortex and lower brain areas like the brainstem, spinal cord, and cerebellum. Interestingly, the midbrain appears to be more involved in voluntary body movement than it is in generating the very basic reflexes we studied in the lower motor system. Recall that those reflexes, like the knee jerk or patellar tendon reflex, are largely generated in the spinal cord. One particular area within the cerebral peduncles appears to be critically involved not only in transmitting commands for voluntary movement, but also in producing them. This area is called the tegmentum, a network of neurons involved in the control of simple body movements and various reflexive and homeostatic circuits. Nuclei in the tegmentum project directly to the basal ganglia, a cluster of neurons just above the midbrain that are key to the production of certain types of behavior. Behaviors driven by evolutionarily conserved motivations, such as hunger, thirst, and sex, as well as habitual automatized behavioral patterns. One specific midbrain nucleus, called the substantia nigra, is very closely tied to the basal ganglia and motor function. In Latin, substantia nigra means black substance, and the substantia nigra is recognizable on brain scans as a darkly pigmented area. This is due to the many clustered dopaminergic neurons in the substantia nigra. These neurons synthesize the neurotransmitter dopamine. They also contain high levels of melanin, the pigment which gives the substantia nigra its classically dark appearance. Dopaminergic activity in the substantia nigra appears to be key for basal ganglionic function, and dopaminergic neurons project directly to the basal ganglia, where they cause secondary motor activation. This midbrain-generated behavioral control is evolutionarily conserved. For example, dopamine has been tied to motivation and habituation behavior even in animals like insects. One key role for the substantia nigra in mediating voluntary behavior is seen in Parkinson's disease, where the death of dopamine neurons in the substantia nigra leads to marked slowness and difficulty in initiating movement. In addition to dopaminergic neurons, select serotonergic and noradrenergic neurons are localized to particular nuclei in the midbrain. Consequently, the midbrain can be seen as both a relay station for receiving and sending information between the hindbrain and the forebrain, as well as serving a modulatory function.